everyone. In this lecture today, I'm going to talk to you about making some dilution calculations in chemistry and also in biology. Okay, I'm going to show you some calculation examples by solving some problems in chemistry and also from biology. Okay, so first this is the really important equation in dilution calculation. This equation is C1 V1 equals to C2 V2, where C1 refers to concentration, starting concentration or the stock concentration, and volume V1 refers to starting volume or the volume of the stock solution, and this concentration C2 refers to final concentration, and V2 refers to final volume. Okay, you have to remember this equation. This equation is C1 V1 equals to C2 V2, initial concentration, initial volume, final concentration, final volume. Okay, so this is one of the most important equation that you must remember. Okay, so now moving to the problem. Okay, let's say that this is the problem. The problem is what volume of a 10 millimolar stock solution is required to make 20 ml of 5 millimolar solution. Okay, so this is our question and we will solve this problem. Okay, so guys, what has been given? We have 10 millimolar stock solution. So 10 millimolar, this is concentration, so it's stock concentration, that means C1. So what is C1? Our C1 is 10 millimolar. Okay, so C1 is 10 millimolar. Yep, so this is our C1 and is required to make 20 ml of 5 millimolar solution. Okay, so to make 20 ml, so this is a final volume, and 5 millimolar solution, so this is a final concentration. So then what has been given? Final volume is V2. So final volume is how much is final volume? 20 ml. Okay, 20 ml is final volume. And what is final concentration? Final concentration is 5 millimolar. So we will write C2, which is final concentration, and that is 5 millimolar. Okay, so this has been given to us, and what they have asked, what volume of a given 10 millimolar stock solution. So they have asked is what volume. So we have to find out what is the volume of stock solution. Okay, that is V1 is what? This is what we have to find out. Okay, so from this equation we have to find out this initial volume. What volume? So V1, stock concentration 10 millimolar, so C1 equals to 10 millimolar, and final volume is 20 millimolar, and 5 millimolar is our final concentration. Okay, so now, so we will apply this equation. So the equation is C1 V1 equals to C2 and V2. Okay, so C1 we know this is 10 millimolar, so 10 into V1 we have to find out, and C2 we know this is 20 millimolar, so 20 C2 is 5 millimolar. Okay, so C2 is 5 millimolar, sorry, so it will be 5 into, and V2 final volume is 20. Okay, so V2 is 20, so it will be 20. Okay, so then V1 equals to, so what will happen? 5 times 20, it is 100, yes, and divided by 10. Okay, so it will be 10 ml. Okay, so what is the initial volume of the stock solution that will be required? That is 10 milliliter of 10 millimolar stock solution is required to make 20 milliliter of 5 millimolar solution. So how we will make the solution? To make the solution, one has to take 10 ml of this stock solution and plus 10 ml of water or any other solvent, and up to 20 ml, right? So this is how the solution will be made. Okay, so final volume will be 20 ml. So this 10 ml you know, it can be water or any other solvent, you know, in whichever the solvent you have to dissolve, you will put that particular solvent. So this 10 ml is from here, from our calculation. Okay, so this is one example, guys, and I will give you more examples to make you more clear. So another question we have is that if 500 ml of 15% volume by volume solution of methyl salicylate in alcohol is diluted to 1500 ml, what is the percentage strength volume by volume? Okay, so guys, what has been given to us? Okay, even though this problem looks a little bit different, but it's easy, guys, don't worry. Okay, so if 500 ml of 15% volume by volume solution of methyl salicylate in alcohol is diluted to, so that means this is our final, this because this has been diluted to. So from what? From these 500 ml of 15% volume by volume solution. So the concentration C1 or the stock concentration is 15%. Yes, 15% volume by volume. And we have our 500 ml. This is our initial volume is 500 ml. Okay. And final volume is 1500 ml, okay, diluted to 1500 ml. So final volume is 1500 ml, V2 is 1500 ml. And what we have to find out is what is the concentration, C2 is what, okay, concentration, we have to find this out. It's very easy, we will apply the same formula. The formula is C1 V1 equals to C2 V2. Okay, what is C1? C1 is 500, yes, into V1 is 15%, so we will just write 15, and C2 is what we have to find out, C2 into V2, final volume is 1500 ml, so we will just put 1500 ml, okay. So now then 500 times 15, so it will be 7500, yes, equals to C2 times 1500, okay. And so 7500 and this 1500, okay, we will divide this both sides by 1500, so it will come here 1500, yes, and C2, okay. So then this cancel and 15 5, yes, C2 is 5% volume by volume, okay. So this will be the concentration of our final solution, okay. So why we wrote volume by volume? Because here it's volume by volume and ml, so this is ml, this is volume by volume, and this is also ml is cancel out, so we are only left with percentage volume by volume, so that's why we wrote this one, okay. So C2 will be 5% volume by volume. Okay, this is another example, guys. Now I will also solve more examples, more problems. So another problem is that you have a 100 ml stock solution of 100 mg per ml ampicillin in deionized water. You want to make 30 ml of 25 mg per ml ampicillin in deionized water. How much ampicillin stock and how much deionized water are needed to make this solution, okay. So these kind of solutions you have to make in a lot of molecular biology experiments, guys. So we will solve this problem. Okay, so what has been given to us? So we know we have 100 ml stock solution, okay, of this much concentration. So our initial volume is V1 is 100 ml. This is our stock solution, and initial concentration, that is C1, is 100 mg per ml. Okay, 100 mg per ml, and is in deionized water. This solution is deionized water, and you want to make 30 ml of 25 mg per ml ampicillin. Okay, so this is okay. So let's see, 30 ml. Okay, we want to make 30 ml. So V2 is 30 ml, right? And this is 25 mg per ml is C2, right? This is C2 is 25 mg per ml. Guys, this 100 ml even though I have written this as V1, but this is not actually V1 because we don't know how much initial volume we will take. Actually, this is total volume that has been given, and from this equation what they have asked, you want to make this ampicillin and how much ampicillin stock and how much deionized water are needed, right? So how much ampicillin out of this 100 ml ampicillin stock, how much ampicillin stock is needed? So V1 is what? Why this is not V1 is because this is the total volume of the solution that we have, okay, 100 ml stock solution, right? And to make 25 mg per ml ampicillin solution, how much volume we need to take, that we don't know, V1, okay, that's why it's what? This will simply be V. So then V1 we don't know, we have C1, we have V2, V2 is 30 ml we have to make, and 25 mg per ml is the final concentration. So we will calculate how much volume of stock solution will be required. As you know, the popular equation C1 V1 equals to C2 V2, and so we have C1 is 100, right, mg per ml times V1 we don't know, and C2 is 25, okay, 25 mg per ml into, we have 30, right, 30 ml, okay. So mg per ml and mg per ml, this cancels out, and so we have 100 into V1 equals to 25 into 30 ml, okay. So V1 will be then 25 into 30 ml divided by 100, okay. So 25 4 times is this, and so it will be 15 by 2 or simply 7.5 ml. So how much of the stock solution we will take, okay. So this amount of the stock solution that will be taken is this 7.5 ml stock solution will be taken, and how much more deionized water we have to add, because we have to make final volume is 30 ml, right guys, from here, okay. So we have final volume 30 ml from here. So then what we will do is that we will put 7.5 ml stock solution, and 30 minus 7.5 which will be 22.5 ml deionized water, okay. So we will mix these two, 7.5 ml of stock solution and 22.5 ml of dewater to make 25 mg per ml ampicillin solution, okay. So now moving to the next and the final problem of today's lecture. It says that how do you dilute 5 times solution to 100 ml 1x, okay. So basically what has been given to us, we have been given the initial concentration, or the concentration of the stock solution is 5 times, and we know that the final volume that we have to make, V2 is 100 ml, and the final concentration, or the C2 that we have to make, is 1x, okay. So then what is how do you dilute it? The question is, how do you dilute it? So basically we have to answer what is the initial volume that we have to take, okay. So V1 is what? We don't know. So to make this dilution, what we will do, we will apply the formula. Our standard formula is C1 V1 equals to C2 V2, and we know C1 is 5x times V1, we don't know how much we have to take, C2 is 1x times V2 is 100 ml, okay. So then this x and x, this cancels out, so then 5 times V1 equals to 1 times 100 ml, okay. So then this will be V1 equals to 100 divided by 5 ml, so this will be 20 ml. So basically we will take 20 ml of this 5 times stock solution, 20 ml of 5 times stock solution, plus let's say that if we are making the solution in D water, then we will take 100 minus 20, which is 80 ml D water to make this 1x solution, okay? Or any other, I mean this this is dependent if we have to take water or any other liquid, that depends in which we have to make this dilution, okay? This is really important. So the answer to this question is we will take 20 ml of the stock solution and 80 ml of the diluting solution, okay? Diluting liquid, whichever the liquid we are taking to dilute this, okay? So this is how we will make 1x solution from 5x solution, right? Okay, sorry, I said last problem, but this will be actually the last problem of today's exercise. So the question is, how much in milliliters of 1% stock solution of certified red dye should be used in preparing 4000 ml of mouthwash, that is to contain 1:20,000 weight by volume of the certified red dye as a coloring agent, okay? So this is our question, okay? So what has been given? What has been given to us? We have been given initial concentration C1. C1 is 1% of the stock solution concentration, and so we use in preparing 4000 ml. Okay, so this is final volume because this is what we are preparing. So V2 is 4000 ml, and that is to contain 1:20,000 weight by volume. So we have been given C2. C2 is 1:20,000, which can be written as 1 divided by 20,000, and I can also convert it into percent into 100. Okay, so basically this and these two zeros cancels out, and which is 1 by 200%. So which is, if we divide 1, so 2.5, okay, so basically 0.0, if it was only 2, it would be 0.5, 0.005%, yep, so it is 0.005%, right? C2 is 0.005%. Why I converted it into percent is because we have our stock C1 in percent, so I converted C2 also in percent so that we can cancel percent and percent, okay? So now it's easy, guys. Now we have already done conversion, so it will be very easy now. I will just separate it out. So we will apply our standard equation, which is C1 V1 equals to C2 V2, and C1, what we have as C1 we have 1%. I will not write percent because, or I can just write it 1% times V1, we don't know how much we have to take, and C2 we have 0.00, okay, 0.005% times 4,000, okay, so it will be 4,000, okay, so which then will be percent and percent, it cancels out, and so we will have V1, 1 times V1 is V1 into 0.005 times 4,000, okay, so which is, then these two zeros, this gone and 1 also gone, 5, so it's equal to 20. I think it will be 20. You can use the calculator. Let me check. Okay, so 1 2 3 zeros gone, and 5 4 is 20. Yeah, 20 ml. So basically we will take 20 ml, okay, 20 ml of our stock solution, 1% stock solution, and dilute it to 4,000 ml, okay, to prepare 1:20,000 weight by volume of certified red dye, okay. So we will take 20 ml of stock solution and dilute it to 4,000 ml to make this much concentration, 1:20,000 concentration. Okay, with this I would like to end today's lecture, guys. Thank you very much for your kind attention. Please subscribe our channel for more content.